Welcome to the latest edition of Gene and Cell Therapy Updates, covering breakthroughs in the past 2 weeks, brought to you by Lucid Quest. Sangamo Therapeutics advanced its rolling BLA submission for ST-920 in Fabry disease, submitting both clinical and preclinical modules to the FDA. The agency agreed that the 52-week mean annualized eGFR slope from the STAR study may support an accelerated approval pathway, potentially shaping future regulatory and payer decisions. Atamyo Therapeutics reported early data from its Phase 1b/2 trial of ATA200 in LGMDR5. Initial results from the first treated patients showed strong sGCG expression, reductions in muscle injury biomarkers, encouraging functional outcomes, and no serious side effects in 4 patients, though follow-up remains limited. Mink Therapeutics and See Further announced a collaboration to advance a PRAME-targeted, TCR-engineered, invariant natural killer T-cell therapy for pediatric cancers. The program will receive non-dilutive funding and support IND-enabling studies as it progresses toward candidate nomination. Capricor Therapeutics received a new regulatory milestone for Deramiocel as the FDA resumed BLA review following a prior complete response letter. The resubmission was classified as Class 2, with a PDUFA target action date set for August 22, 2026, re-establishing a defined regulatory timeline. Ultragenyx announced that its partner Grit Biotechnologies received FDA IND clearance for GT307, a gene-edited tumor-infiltrating lymphocyte therapy. The milestone enables initiation of first-in-human clinical trials and reflects continued expansion in engineered cell therapy platforms. Sarepta Therapeutics has begun screening and enrollment for Cohort 8 of the Endeavour study evaluating Elevidys. This cohort will assess peri-infusion sirolimus in non-ambulant Duchenne patients to reduce acute liver injury risk while measuring dystrophin expression. Ultragenyx reported that its gene therapy, DTX301, met one co-primary endpoint in a Phase 3 study in OTC deficiency. The treatment reduced 24-hour plasma ammonia levels by 18%, compared with placebo at 36 weeks, with further endpoint data expected later. Solid Biosciences shared additional Phase 1/2 data for SGT003 in Duchenne muscular dystrophy. Findings included robust microdystrophin expression, restoration of associated biomarkers, improved muscle injury markers, and early signals of cardiac benefit as the program advances toward Phase 3. Stay ahead in gene and cell therapy research. Like, share, and subscribe for our updates. Visit lqventures.com or email us at info@lqventures.com for